Yeah, we're going to start off the program with an overview of the vegetative and reproductive cycles of grapevines, because I think if you have a basic understanding, get everyone kind of on the same page there with the reproductive and vegetative cycles, then I think you'll have a really good understanding of how grapes are grown and managed and why certain cultural practices are done. So what we're going to do is introduce 3 integrated annual cycles of the vine: vegetative growth, cluster initiation, and fruit growth and development. I'll just briefly touch on fruit growth and development because I think my colleague Ashraf El-Karimi is going to discuss this in much more detail in the talk right after mine. But I'll just kind of dip our toe into it so you'll be ready for Ashraf's talk. And then I also want to demonstrate with a few examples how the environment and cultural practices affect these cycles and thereby affect fruit yield and quality. So we'll just kind of get a basic understanding of how the grape grows and how it produces fruit and what are the environmental factors and the things that people do to grapes that affect these things. Well, it's too bad that for whatever reason, the projector is clipping off the top and bottom of my slides. So if you were able to see the whole slide, then what this would say is vegetative growth cycle, okay? And so we are just going to kind of walk you through the vegetative growth cycle here. And where we're at right now is we're more or less in this dormancy period. And at some point during dormancy, people typically prune the vines, and they do this to manage their crop, and we'll, I think, by the end of the talk, you understand why that's so. And in maybe 3 weeks or so, you'll probably start to see bud break. Down where I am located, down in the San Joaquin Valley, I was out walking around the other day through some of the vineyards where I work, and I noticed that some of the vines kind of in more warm areas are starting to show some signs that they're getting close to bud break, but probably more like in 2 to 3 weeks. After bud break, the vines grow relatively slowly to start because the weather is cool, but then soon they'll get into this rapid growth phase. That usually, they really reach their peak in terms of their growth rate right around bloom. Shortly after bloom, we'll enter this period called berry set. And then after berry set, the vines will be at full canopy. And then they'll start to develop a periderm, which people refer to as cane hardening, basically kind of getting lignified shoots. And after that is when harvest typically occurs. And then when the weather turns cold, which in the last 5-6 years it seems like is getting later and later, but eventually at some point during the winter the leaves will fall, and then we'll be back to where we are now and ready to prune.
What I was, where we're at anyways is still in the dormant season, okay? We can at least agree on that, and here are some people pruning the vines, and this is partly just to show you kind of what they look like. You've probably already noticed, like I said, driving over to the hotel, what some dormant vines look like, and here's kind of the before and after of pruning. They're leaving a few nodes, and we'll talk about the importance of those nodes they're leaving behind starting in the next slide here. So if we were to, now that this slide is all scrunched down, and so you can see the whole slide, it's probably harder to see the buds than it might have been before with a little bit larger image size. But what they're probably doing, and my eyesight is not as good as it was when I was younger, so I cannot be sure, but I think if we could see this a little bit better, then probably you'd see like each of these little sticks, or like a 1-year-old piece of wood, basically, that they're leaving behind after pruning. These people are what we call spur pruning these vines, and the difference between a spur and a cane is just the length of the 1-year-old wood that they leave behind. So a spur is a short length that only has like 2 nodes on it, and a cane is a long length that has more than 2, maybe 15, or I'll show you some later that have like 20 nodes maybe. And so they're leaving just a couple of these nodes behind and removing all the rest. And here's like a blown up version of one of the nodes. The space in between two nodes is called an internode, and this is the node. So the internode is between nodes, and this is the node. And within the node, which a lot of people refer to as a bud, the reason why we call it a node is because if we just called it a bud, it's actually like a compound bud. So if we were to peel away these scales that are covering it, we'd see there's actually two or three buds inside that structure there. But anyways, that's what we're seeing right now in the state of dormancy. If we look at the shoots of the vine, we see a lot of these. This is what the shoots look like. They've got these bare nodes and the lignified canes. If we go out there again in a few weeks' time, we'll start to see that they've shed their bud scales, and the buds are starting to swell. And what causes this transition from dormancy to emergence, at this point, there's actually 3 different stages of dormancy that these buds go through. As we'll see shortly, these buds are developed on when the shoots were green the year before. So when this shoot was green the year before, that's when these buds were formed. And during that season when the shoots are green and it makes these buds, they are kind of held in place. They're prevented from growing by hormones that the growing shoot tips produce. And that kind of represses the growth of these buds, and that's a kind of dormancy called correlative inhibition. But then later, as we get into the tail end of the summer and into autumn and certainly by winter, they're in a deep dormancy, and they cannot be released from that deep dormancy without some chilling. So cold temperatures actually act on the buds, and it causes some physiological changes within the bud that once enough chilling has occurred, then they can actually be released from this deep dormancy. And then they enter like a third kind of dormancy, which is like environmental dormancy. So then by this time, all of the buds in California have achieved enough chilling for the most part, except in maybe a few particular regions. But in general, they've received enough chilling. So the only thing keeping them from growing now is temperature. So once the temperatures start to rise sufficiently that they can grow, they will grow. And that's where we're at right now. They're being held in check now by cold temperatures. But depending on the variety, once the temperatures reach a certain point, they'll start to grow. And when they first start to grow, they start off a bit slow. There's a lot going on these little shoots. They're starting to develop the clusters that they're going to have more fully, and the leaves are starting to expand, and the temperatures are relatively low still. So all these things kind of combined lead to relatively slow growth rate in the beginning, which is good because usually around bud break I go through this period of panic, like feeling like I'm not ready for the season to get started, and so I kind of appreciate the slow growth rate. But it won't be long before they start really kind of picking up steam. And when they reach about this size, that's when you can start to see quite clearly the clusters start to separate from the shoots. And I came into this job from other kinds of plant systems, and I didn't really know much about grapes when I started, and I had never really seen grape flowers until that first year on the job, in person anyways, and I was really disappointed because to me they look like broccoli or something along those lines. I mean, they're certainly not exotic looking in any way, but I've come to appreciate them. They smell really good. They have a very unusual and distinctive smell, kind of like an herbal shampoo or something. But how we're going to tie in this shoot discussion with this shoot discussion is going to tie in with the fruiting cycle discussion here. And this slide is kind of the introduction to that because I want to point out this structure here in what we call the axil of the leaf. So here's the leaf blade. Here's the petiole, which is the stem that attaches a leaf to the main stem. And in the axis here is what we can see is a lateral bud, or sometimes called a prompt bud. You can see a little shoot growing out right there. And the reason why they call these lateral buds prompt buds is because they grow out in the year that they're formed. So in the axil of every leaf, there'll be at least a small little lateral bud pushing out at every axis. But these lateral shoots, depending on the variety, depending on the environmental conditions, for example, how much water or fertilizer they get, also the temperatures and how much fruit that they have on the shoots and so on, can control how much growth these lateral shoots make. In some cases, this is about as big as they get, and then they'll simply fall off. But it's really important, even this very small, short length of growth, and I'll show you why. Because here is another, this isn't the greatest picture because it depicts a lateral shoot right here next to the cluster. And a lot of people, I've realized a lot of people's attention goes to this cluster. But just forget about the cluster right now. And here's a leaf, and here's this lateral shoot. And right at the very base of the lateral shoot is this kind of enlarged bud or node right there. And that is actually the structure that is going to become, if it's left behind over the winter, that will be that dormant node that I showed you before. So it's really kind of deceiving because when you look at the picture of the dormant node there, it looks like it really belongs to this shoot, right? But it's actually the first basal bud of that lateral. Okay? So here's an example of a variety that had very vigorous lateral growth. You can see like what we call a woody lateral, one that has really developed quite extensively and actually lignified along with the main stem, in contrast with maybe that one before where perhaps that's as big as they're going to get, just a little couple millimeters. So depending on how the vines are managed and also depending on the variety, you might see shoots like this, all these laterals. And the laterals themselves sometimes can form fruit. But what's really important about the laterals is they contribute a lot of leaf area to the vines, and that can be important for productivity and capacity of the vines. Typically, if you had something like this, you'd probably be at least pruning all these lateral shoots off in the winter. Here, this picture probably taken around bloom time. You can see the canopy is getting pretty big, and at this stage of the growth of the vine, they're at their maximum growth rate. So these things are extending very fast. But as soon as the fruit sets, if the vines are being managed in kind of a reasonable way, then usually the growth rate begins to slow quickly after that. And this is an old-time photo, but shows you, anyways, what a whole vine canopy might look like. Maybe not here, but somewhere in San Joaquin Valley. And then once we start to get after harvest and with the onset of shorter days and cooler weather, then typically we start to see the leaves start to yellow, and there's no more new growth. And this is a kind of an autumn time picture, and you can see the leaves are all yellow. And this is yellow color is really the normal color for senescing grapevine leaves of most of the grape varieties that we think about. When you see red leaves, that's often an indication of a virus disease or something wrong with the vines. So just kind of a pale yellow is sort of the normal appearance of senescing leaves. After leaf fall, again, this is maybe not your typical Napa vineyard, but a very vigorous vineyard in the San Joaquin Valley. But I like this picture because it's part of a series. I'll show you the second shot just to reinforce how much material is coming off of the vines sometimes at pruning. And so you can imagine all of these shoots have those nodes like we showed you before. All of those nodes have shoots on them. Many of the shoots have clusters on them. And so you can imagine how many shoots and clusters could potentially come out of these vines if they weren't pruned, and it would be too many. And so pruning, this pair of slides here mainly just shows you how much potential crop adjustment and vine capacity adjustment occurs at pruning. Obviously, a lot of vines in a lot of places, even in the San Joaquin Valley, don't look like this anymore. But still, we prune away an awful lot of potential productivity each year in order to get the fruit quality that we want and to manage the yield for the needs that we have. So that's the vegetative cycle, and I already kind of cheated a bit and dropped in a few ideas about the reproductive cycle, but we didn't really get into it in depth. And so now I'll kind of focus much more on the reproductive cycle, and we'll show a lot of the same slides that I did before, but now really keying in even more on how that fits in with the reproductive cycle. So the reproductive cycle of grapevines is a 2-year process. In the first year of the process, flower clusters are initiated and they partially develop on microscopic preformed shoots within the buds on green growing shoots. Okay? It's like a really huge mouthful. I need to figure out how to edit that better. But then in year 2, so first year, what I'm trying to say is that the clusters, the flower clusters, the clusters of grapes, basically the primordial clusters of grapes, they're made, next year's grapes are going to be made this spring. The clusters of grapes we'll have next year, they will be initialized this spring. And they'll form in those buds that I showed you before that are being formed on these green growing shoots that will emerge in a couple of weeks. Now, those buds will form these microscopic shoots with already the clusters attached this spring. Then, if you don't prune them off, then they will continue to form on those little shoots. And in year 2, when those shoots emerge from the buds, from the nodes, then the flowers will finish developing and bloom will occur and then fruit set and berry growth and maturation. So from the time that the cluster is initialized in the spring to the time when the fruit are harvested the following year, that's like 15 months or so. So those grape berries have, it takes them longer to develop maybe in some ways than a human baby. So let's look at that in like a diagram kind of a way. Year 1 in the developing primary bud or node, the clusters are initiated, and then some rudimentary development occurs, like the rachis elongates a bit. And to say rachis elongation, remember you're going to need a microscope to see this, but it is happening. And then some flower initiation perhaps occurring toward the end of the winter period. And then in the second year, this is all occurring within the bud. And then in the second year, when the shoot emerges from the bud, flower part differentiation will occur, and then bloom, and then pollinization, fertilization, berry set, berry softening, harvest. And I think it's important to think about these things. The reason why I'm telling you this information is because growers will come up to me and they'll say, "Oh, my yields are lower than expected," and I'll say, "Well, what is the problem? Do you have, how many clusters do you have on a vine?" I would start off with that because that will tell us something about that might have happened not this year but last year, right? But then, okay, what if they have a normal amount of clusters but they don't have very many berries? Well, then we know it happened this year, So if you know a bit about this whole process and that it takes 2 years and what are the things that can go wrong in the 1 year versus the next year, and then you start to think about what is happening, what are the components of your yield, how many clusters did you have, how many berries did you have, how big were the fruit, that will inform what the problem is and help you manage your vines better to get the results that you want. So we're back to this good old compound bud again, talking about the reproductive cycle. And there you can see I think in this other slide we might have been able to see the very first sign of a cluster. When the cluster first is apparent, you have these special leaves called bracts that cover up like parts of the branches of each cluster. And so it looks as if it only has a couple of flowers on it or something, because the bracts almost look like the flowers, when you're just kind of looking at them with your normal vision, at least my vision. But then as the branching develops within the cluster and they start to kind of emerge away from their little protective bracts, then you can really see the full extent of the clusters that you have. And here we're approaching bloom. There's the lateral prompt bud that we talked about. And this is, I cheated and told you about this in the vegetative cycle, but here's a good review of it. The dormant bud arises from the most basal node, okay, so of a lateral shoot. So here's the primary shoot. Here is the lateral shoot. And then its most basal node, the one nearest the base of it, is what is going to form that dormant node. And within that dormant node, there'll be special meristematic tissue that we call an enlage that develops either into a shoot, a tendril, or a cluster primordia in the spring. So right now, or at least when I say right now, I'm talking about in the context of this picture, there's little shoots in there. They have these little enlages, enlage is singular, enlagen is plural. They have these on those little microscopic shoots. And they're kind of sensing the weather around them and also other biological clues like hormone balance within the shoot and also other factors that we'll talk about. And they're deciding whether they're going to form a tendril, a cluster-like tendril, or cluster, all these variations. Some of them, sometimes you'll even see a shoot come out of those, although it's kind of rare. And so that's all happening within that structure. So if we go and really bore into this little bud here, like we did in this slide here, we just kind of tore into that bud to kind of take a look at what's happening. Here is the apex of that microscopic shoot. These are leaf primordia. Okay, so these are going to become leaves the next spring when they have the opportunity to grow and emerge from that bud. And here's one of those enlages. And that enlage is like uncommitted primordia. So it could become, again, a tendril, a shoot, or a cluster. And there's kind of like, it's not like an on-off button or a three-way kind of switch, like shoot, cluster, tendril. It's sort of like a continuum. So I've seen clusters that have a shoot growing out of them. I've seen a tendril with like one or two flowers on it, and everything kind of in between. And depending on the different cues that this enlage gets, it's making its decision to do one thing or another. But once it makes its decision, then it's set. And certainly by the end of the summer, in those buds, next year's potential crop will be set. You could look at them if you had the ability to and figure out what you had. So what regulates what happens with this enlage? Or what are some of the environmental regulations that affect bud fruitfulness? So bud fruitfulness, that's just how many clusters are there coming out of the associated with this bud, or that bud. So we could actually dissect these buds and count how many clusters were there, or we could wait until the following spring when they grew out and we could count how many clusters. That's what bud fruitfulness is. So it's been recognized for a long time that light and temperature are both positively correlated with bud fruitfulness. So the warmer temperatures, more light, you tend to have more clusters initiated at each bud. So here's an example of that with some data. So here's primordia per bud. These are cluster primordia per bud. And here is light intensity. These are different varieties, Rieslings, Syrah, Muscat, Thompson. And you can see that even though the slope of these lines is different, they're all positive. So you get more primordia with higher light. So So that's been recognized for a long time. And it turns out that also temperature is positively correlated with the primordias per bud. So you'll tend to have more cluster primordia as the temperatures are warmer during that initiation period. So again, in the spring, these Enology are committing to becoming whatever they're going to be, like right around bloom time of this year. So like in May, basically, in like May, June, depending on the variety, maybe even extending a bit longer. That's when those little buds, the shoots inside those little buds, are starting to make their decisions. Okay, cluster, tendril, whatever. Shade. So we showed that light promotes fruitfulness. Well, shade can decrease it, and here's some data showing that. Here's number of days of shading of shoots from 0 to 45 days. And there's two things we're showing here. One is the bud fruitfulness, and you could probably guess because we showed you before that light was correlated with higher bud fruitfulness. So shade is correlated with lower bud fruitfulness. And it looks like 15 days is enough, basically, and more than that doesn't further decrease it. But there's also here something I didn't point out before, which is bud necrosis. So for these buds, when if you want to produce a good crop, you need to not only have enough clusters initiated, but you also need to keep those buds alive through the course of the summer and over the winter, and so that they can emerge in the spring. Right, the shoots can emerge from them. And so if you have bud necrosis, that's like the death of the buds. Well, that is increased by shade. So shade increases bud necrosis, which means fewer of the buds survive. And the buds that do survive have lower fruitfulness. And so you're kind of getting two benefits from sunshine, or two negatives from shade, however you want to look at it, with the survival of the buds and also with how fruitful they are. This I just showed you. I'm just throwing in there just to show you these. This is just a cross-section of a cane with xylem rays, starch in them stained with a blue dye, just to show the accumulation of carbohydrates in this case in the cane, and how much carbohydrates that the canes have are by the end of the growing season are associated with good things like better bud survival and also higher fruitfulness. And also even the buds themselves, so these total non-structural carbohydrates, these are like sugars and things like that that the buds can use, the structures can use for energy and so on. And here's bud fruitfulness. And you can see a positive relationship with bud fruitfulness versus how much sugars and non-structural carbohydrates they have. So having better reserves of carbohydrates is good, and that makes sense with the light and the temperature relationships that we saw before. It seems like there is actually a direct, there are some studies that show just a direct benefit of warm temperatures, but also the warm temperatures probably co-vary with the light. When there's more light on the canopy, probably the tissues are warmer, and the light certainly is what's responsible for the more carbohydrates and the more starch and so on, which also helps the clusters develop, the onlogans commit to making clusters. So when is all this happening? I've mentioned it kind of a little bit before, but now we can actually see a graph. Here's bud fruitfulness percent. This is what percent of buds have clusters, and this is time. And here you can see we're going kind of around from bloom, berry set, veraison. So it's just increasing all the way up to veraison. So around bloom, before and after bloom, basically, but certainly by veraison, most of those cluster primordia have decided to become clusters or not. So that's just when these things are making their decisions. So certainly, I said by autumn in the slide, but in the previous slide it was showing veraison. So certainly by midsummer to autumn, the number of clusters each shoot could bear, and by shoot, again, I really mean these little teeny shoots that are inside these buds. they've already made their decisions. They're committed. So we have George is in the audience. I'm not sure if he's giving a talk or not. There he is, the man who's the bud dissection master. Here he is holding a bunch of canes. Okay, a cane is just a long section of lignified primary shoot, right, from the end, collected from the end of the season. And in the San Joaquin Valley, a lot of our varieties are cane pruned, so we leave long sections of wood, not just the short sections like you might leave up here, which we would call spurs. So we leave canes in a lot of varieties, and so he's brought a bunch of canes in, and he's going to dissect some of these buds. He went out there and randomly sampled some canes. He's going to dissect the buds on the canes to come up with pruning decisions, because those buds already have their clusters on them. You just can't see them unless you dissect them under a microscope. And if you do dissect them under a microscope, which George does every winter, then he can actually come up with a report. Okay, this is our average bud fruitfulness. This is, kind of what we're getting in positions, let's say 1 through 15 or something. And that can tell us how many canes we want to leave and also how long they should be. But that could be beneficial also to spur pruning, because you might collect some spurs and dissect those and decide you need to leave more spurs or you need to leave longer spurs or whatever the case may be. So here's what you're doing here. You're dissecting those buds to see the potential number of cluster primordia, inflorescence primordia, same thing. There's George with the microscope, and this is kind of what he's seeing, a cartoon of what he's seeing. So here's like a profile view of the bud, and he is kind of shaving the top off the bud sequentially until he gets into these shoots themselves that are within that bud. And then this is sort of like a slice through the bud that was flipped over on its side there. And you can see 1, 2, 3 shoots, and you can see there's 1, 2 cluster primordia there. And so he can just go and do that and count his buds. So here's a graph he made from those canes that I showed you before of potential bud fruitfulness. Here's the number of cluster primordia from 0 to almost 2. And node position, these were from canes. So the most basal node in the cane, and then the first, what we call a count node, and all the way through like 13. He went 13 nodes out. And this is on average what he saw. And this is pretty typical of cane pruned varieties, that they have low fruitfulness at the first couple of nodes, and it increases quite a lot, usually reaching a maximum either in the middle or the end of the cane. And that's why some varieties are cane pruned instead of spur pruned, because if we spur prune this variety, then we'd only be getting about 0.2 to 0.4, a quarter to a half of a cluster on average for each node, whereas these more apical nodes, we're getting far more clusters per node. So that's why you would potentially consider cane pruning instead of spur pruning. Get a much bigger crop that way on certain varieties. So now with your information, you can decide how much of this stuff to prune off. You could guess if you did bud dissections over time. They're not 100% accurate for various reasons. You get to get a feel for how well they work in a particular vineyard over time, so it's good to do them kind of continuously. But certainly you're going to get some kind of feedback. You'll have an idea of how many clusters you might potentially have, depending on how many nodes you leave. And so you can decide how much pruning to do, and there you go. That's what you have left. And we already discussed this, so I won't belabor it anymore, but sunlight promotes cluster initiation and development and bud survival. The number and potential size of the clusters on the preformed shoots is positively correlated with light, temperature, carbohydrate accumulation. In early summer when the clusters are initiated, shading reduces the cluster number and potential size and promotes bud necrosis or death. And so oftentimes we recommend, if you're cane pruning, to prune sun canes versus shade canes, because the sun canes are the ones that kind of are overarching on top of the others, and they were the ones that had the better sun exposure when they grew out, and so they're going to have more shoots at each node that have clusters as compared to the ones that are more in the interior. And The reason to think about all this stuff, I mentioned it right from the very beginning, because then you can make some understanding about the clusters that you're going to have per vine, the berries per cluster. So, if you're putting together the entire yield for your vine or for your vineyard, it would be clusters per vine, berries per cluster, berry weight. All those things together end up being your yield for wine grapes anyways, or for table grapes. For raisins, kind of a specialized crop, those are dried, and the sugar per berry is also important. And if you're thinking this is true on a vine scale, and then if you're thinking in a vineyard scale, I guess you'd also want to know how many vines per land area that you have as well. So now we're in the year 2 of that vegetative process. Remember, it's a 2-year cycle. So now we're in year 2. The things that could go wrong in the first year we discussed. Now, the clusters that were initiated last spring, they're going to complete their growth this spring. So the clusters that we see this spring, and maybe we might be able to actually see them in about a month or a month and a half when we'll get our first glimpse of them, they were formed, remember, last spring on the shoots that were growing then. And now they're going to finish their development, these clusters that were formed last year. And during this time, cluster development can be directly affected by the weather, insects, nutrition, and other factors. Before there were mainly environmental and biological factors kind of affecting them, right? Whether they're going to become a cluster or not, and how big they might get, and whether they're even going to survive for the winter. But now they're going out into the world, all exposed, and so they have other kinds of things that can impact them. Here's that kind of broccoli look that I was telling you about before. Kind of looks like broccoli rabe, maybe, Rob. Now, grape flowers are kind of weird, I think, because one of the things that makes them distinctive, you can see those yellow kind of specks. Those are all the anthers that have pollen on them. And you can see when the flower has bloomed, because that's when the anthers are exposed. Before then, they're tucked inside and they're covered with the petals. The petals are green colored. They don't really look like a petal. And the other weird thing about them is that these petals are fused at the tips. And so they actually come off the flower at the base. So the petals remain attached to each other at their tips. The base is what breaks off, and it comes off like that, and it looks like a hat. And so we call them calyptros, which is Greek for hat or cap. And you can see here's like a petal, and on this flower it's just come off. And you can see they're still attached to the base, but the tip, but the base of the petal is detached. Here, all of the petal bases have detached, but the anthers are still covered with the cap. And then now the cap is off. And the action of the cap coming off generally seems to pollinate the flower. Bees do like grape flowers, but they are not necessary to pollinate the grapes. So the grapes will be pollinated whether there's bees around or not, and a grower won't bring bees into the vineyard like, for example, they would in an almond vineyard. It's not necessary. Although there are reports that pollination is better when there's bees, but it's sufficient without the bees. Anyways, here's the anthers. There's the stigma, which is the female part that's sticky and collects the pollen. And then the pollen will germinate there and grow down into the ovary and fertilize the ovules. And the number of berries per cluster and berry size will depend in part on pollination success. So grapes don't require insect pollination, but poor weather and mineral nutrient imbalances can substantially reduce set. So if you go and there's a lot of reports out there where they've gone and dissected berries and found that the berries are usually smaller if there's fewer seeds. So the more seeds, basically, it's because there's better pollination. And if it's not pollinated at all, then those flowers will tend to fall off. Micronutrients sometimes are limiting, and zinc is needed for the pollen tube to grow properly, and so fertilization is impaired when vines are zinc deficient, which is something that we have problems with in the part of the state where I'm at. Plant growth regulators, you don't really see this in wine grapes, but in table grapes, growers use certain plant growth regulators to enhance berry size, and so that's obviously going to affect your crop components, your yield components. And I think I'm going to leave it there. I was going to talk more about flowering and fruiting, but I know Ashraf El-Karimi is going to do that. So I don't think I have this particular presentation on SlideShare right now, but maybe but it sounds like you guys are going to get a copy of it anyways, but I have other presentations that I generally put on SlideShare. So if you want, it's free. You don't have to be a member. Just go to slideshare.net/viticulture, and I generally have presentations up there. This is my email address if anything didn't make sense or you want more information. And if you're on Twitter, so am I, and I'm at Grape Tweets. And so that's it. And sorry about all the problems we had in the beginning, but I hope you enjoy the rest of your day, and I hope it doesn't rain for you guys. Too bad. Well, I know it's going to rain for you, but I hope that you come up with an alternative plan that is still useful to you. So enjoy the rest of your time. And okay, yeah. So you do see sometimes 2 or 3 shoots coming out of those dormant nodes rather than one. And where you most often see multiple shoots coming out is when you prune the vine very severely. So the vine had a higher capacity, let's say, and you pruned it very harshly, so you didn't leave very many nodes after pruning. The fewer nodes that you leave, I think the more frequent it is that you get more multiple shoots coming out of the node. The other thing is if you have some injury to the primary shoot, then you will sometimes see the second shoot or the third shoot coming out. So those are the things that I think really promotes multiple shoots per node, is basically a high capacity relative to the amount of nodes that the vine has, and then also kind of environmental conditions like bud necrosis of the primary node and things like that. The primary shoot usually is the most productive, but you can often salvage a crop with secondary, with clusters that might be on the secondary shoot. The third shoot usually doesn't have much. And so I've worked in a table grape vineyard one year. One of my studies was in a particular part of the San Joaquin Valley that's very prone to hail. And they had an intense hailstorm soon after bud break, and it knocked a lot of the primary shoots off. And I was wondering, oh man, we're really going to have almost nothing at the end of the year. But the secondary shoots came out. And then with the fruit that they had, plus also because those vines are big, strong vines, and now all of a sudden they have many fewer shoots than they would originally have, a lot of the laterals had clusters too. And at the end of the year, there's quite a lot of fruit there. So, yeah. Yes?
If you allow your vines to overproduce, that is, if you've got too many clusters, a lot of carbs going into those clusters, does that then reduce the number of carbs that are there to help those buds for the next shoot develop?
It can, yeah. And so the vines will kind of regulate themselves. If you don't do the regulation for them, then they will do it themselves. And so they will often have, you will have many, many clusters for sure. If you like that one vineyard that we showed you the before and after pruning, you know, if you left that thing unpruned, it would have many, many shoots. But the actual bud break percentage might have been lower than it would have been if you had pruned it. Because like the previous question, you know, when are you going to get all the shoots coming out, basically? That's because you prune it and you kind of give it no choice. It allows only a few shoots to emerge from, a few buds to emerge from. If you leave it the option of many buds, it will not emerge as many shoots on a percentage basis. But still on a number of shoots, there'll be far many more shoots on an unpruned vine. But then what you'll end up getting is poorer development of the cluster. Because remember, I showed you a lot of the cluster development actually occurs in that second year. soon after bud break, they start differentiating, getting more branches, and the flowers form and so on. That will be less developed. And so the clusters will be smaller in general. And that's what you'll have, a lot of smaller clusters with poor fruit set and shorter shoots.
On a brand new vineyard, how do you determine whether to train the vine for canes for
On a brand new vineyard, so if it's on a brand new vineyard, then I think your first question would be what is the variety, and then find out some information on the particular variety. And there's probably a generally accepted practice. Some varieties are more fruitful on basal nodes than others, and they're sufficient to spur prune. Others are well known to have poor fruitfulness at the basal nodes and be better if you cane prune them. But there's not really any hard or fast rules, especially for a home vineyard. You can cane prune if you prefer. There's different ways of cane pruning, and so you can almost use the canes as like a replacement for a permanent structure that you might otherwise have, like a cordon. And so as for a home vineyard, it's less critical. For like a commercial situation, you would probably know that this variety is best grown this way.
I learned a little bit about short day and long day plants. So with grapes and temperature being the primary reason for bud break, are they a long day plant? Would that be correct?
I didn't really pay too much attention to that topic when I was thinking about this. I wasn't expecting that question, but I don't think grapes are as responsive to day length as other plants are. Is my understanding? Is that?
That's correct.
Yeah. So that I think that there's a weak response to day length by grapes, but it's not that classical day length kind of response. And grapes are grown in tropical areas that have very uniform day lengths, and they grow fine. Yeah.
Regarding Central Valley wine grape growing, can deficit irrigation increase the amount of light and bud fruitfulness on various varieties?
Yes, there's different ways that you could manage your canopy to prevent a very overly dense canopy and too much shading. And one of the ways that you could do that is through careful irrigation and fertilization to not have excessive shoot, bud bursts, so you have too many shoots, like we talked about in the previous example, and that the shoots don't get too big, you have too much lateral growth. So you can there's like environmental kind of breaks you can put on the vine, what we call a cultural practice, or there's also things you could do if, let's say, we have a lot of rain, like this year. The vines are starting off with a full profile of water. They grow very rapidly on their own. That those shoots are already preformed, remember, at least the first maybe 12 nodes or so. And so in the beginning, it's just like expanding its potential, basically. And so one way you can kind of put the brakes on that is just try to have less water. But if it rains a lot like this, then like it is this year, then you may have to do some other means to kind of gain control of the vines. So, for example, my colleague here, I think he may talk about it later, Con, has done a lot of work with canopy management. We're actively managing the canopy. So you couldn't really necessarily control it sufficiently naturally, and so you have to go out, go back and respond to it and take some leaves off or take some shoots out, either by hand or mechanically. And those practices will enhance your bud fruitfulness, whether it's controlling the growth by irrigation management or if you're controlling the growth by hand.
Follow up on that question regarding the water potential. How far can you go without damaging bud fruitfulness? If you have also a lot of water, and then you go to 13 bar, or you say, okay, we can't put the water on now because we allow water to maximize fruit.
I think we're going to have an irrigation session, right?
Yeah, we're going to talk about
Andrew's going to talk about
fine water. Yeah, so.
Yeah, I'm going to want to steal a thunder from someone else who's more of an expert in that area anyways. So.
But if he doesn't answer your question in his talk, then you can ask him. Then, yeah. Okay, one last question, then we need to get on to our next speaker. Go ahead.
When you're talking about canopy management by hand, that's just suckering that you do, like early season suckering to get.
Yeah, we're leafing, like removing the leaves in the fruit zone. And
Exactly.
Thank you.
okay, thanks.